welcome to my talks. And here today the video is on linkage and recombination, standard 12th bio. Okay, so last time we did chromosomal theory, and this time we are going to start with linkage and recombination. So again, the thing must be what is linkage and what is recombination. Linkage is because we are doing genetics, so here I'll talk about genes. So when two of the genes get linked together, that is called linkage. Which two genes, where, we are going to discuss in the topic. And recombination is new characters. When we have new characters, when two different genes come together and the new characters which we have, that is recombination of characters. So recombination. Now here what happened, children, what we did, Mendel's experiments, okay, on genetics. So Mendel gave law of dominance where we saw 3:1, okay, the first one, that is monohybrid cross, and we also did dihybrid cross where we got 9:3:3:1 ratio, taking two characters at one particular time. Now based on that particular experiment, Morgan also conducted quite many experiments. He conducted experiment on drosophila. Drosophila is a fruit fly. Okay, so he took drosophila and he conducted dihybrid cross. Okay, and then what did he do? Okay, he tried to study sex linked genes. Okay, as I was talking about linkage, so here he wanted to study which are the genes which are linked with sex gene. Now children, earlier if you remember, I had told you that we have 23 pairs, total 23 pairs of chromosomes. Okay, out of these 23, we have 22 pairs. Okay, these are pairs I'm talking about. Okay, this is pairs. So 22 autosomes. Okay, and this one pair is sex chromosome. Now only one pair, it means two chromosomes are responsible for deciding the sex of the organism, whether it is a fruit fly or it is a lizard or it is human being, whatever it is. And other 22 pair of autosomes, that decides the characteristic of the other parts of the body, the other characteristics. Okay, maybe the color of the skin, maybe the color of my hair, maybe the way I talk, the way I walk, okay, the shape of my nose, everything. So all that, 22 pair of autosomes and one pair of sex chromosome. Now here there are some genes which go along, which get linked with this sex gene, sex chromosomes. Okay, and those characters, that together is known as sex linked genes. Means along with the responsibility of deciding the you can say the sex of the particular organism, okay, it also carries some of the other genes responsible for some other factors linked with it. Okay, so what did he do? What did Morgan do? Morgan took yellow bodied, white eyed females, okay, those drosophilas whose body was yellow and the eyes of those drosophila fruit flies was white. Okay, and he crossed it with brown eyed. Here also, like dihybrid cross, okay, what we took, the shape of the seed and the color of the seed. What was there? Round and wrinkled, and then the other one green and yellow. Okay, so here also two factors he has taken. Yellow body, here he took brown body, here he took white eye and red eye. Means two characters, dihybrid cross he did. And he wanted to come to the same conclusion as Mendel got. Okay, so what he did, whatever he got in F1 progeny, okay, F1 progeny what he got, he self-crossed it. It means there what we had got, capital T, small t. So when we cross it, capital T, small t. Okay, and this is what we got. So finally, this is one unit cross, dihybrid cross, you took it together and then we found the dominant characteristics present in F1 hybrid. So finally here what he did, F1 progeny ko intercross kiya. So when it was intercrossed, he did not get the ratio which Mendel had got in his dihybrid cross of pea plant. Okay, so there he got 9:3:3:1 ratio. Here Morgan did not get 9:3:3:1 ratio. Okay, and when he tried to find out what was the reason behind it, he saw that the two genes in dihybrid cross are present on the same chromosome. On one particular chromosome, both the genes are present. Okay, both the genes responsible for the color of the body of the drosophila as well as the color of the eye. Okay, they were present on the same chromosome. And then what happens? The proportion of the parental gene combination means there won't be any variation. So sexual reproduction का कोई मतलब ही नहीं होगा. What is the reason, or you can say what is the main aim of sexual reproduction? The main aim of sexual reproduction is variation. So here in this case they will be joined together. कैसे होगा? It will be like this. It will be present. Suppose this is a chromosome, it will be here. So it will go along with the chromosome. This will not get separated, and when it doesn't get separated, there won't be any variation. Parental combinations will be seen at the maximum. Non-parental combinations will not be there. Okay? So finally, what happens, children, over here? This combination that the genes present on the same chromosome and more of the parental combinations are seen. Okay, this is much higher in this case, and this is what is linkage. Now what happens, that I'll tell you. Okay, so there what we had, let me explain with the help of seed color. There we had capital R and capital Y. Okay, so here segregation, we did segregation. Here segregation does not take place. It means the two chromosomes responsible for two different factors, they do not get segregated, they do not get differentiated. Till they do not get segregated, they will not be able to express themselves independently. They'll go hand in hand. Okay, so here this round and green seeds, okay, the yellow seeds, it will be going hand in hand. There won't be any variation. It won't be like round and green won't be there. Okay, round and green नहीं है. Suppose अगर combination आपका है, suppose आपका combination है capital R, small r, capital Y and small y. तो segregation होगा, what it will be? It will be capital R, small r, capital Y and capital Y. And when we have the combinations, when we have the genetic compositions, okay, how it will be? There will be four kinds of seeds. So capital R, capital Y, once with this, once R will go with this. The other one will capital R, small y. Once it will go with this, small r, capital Y, and once it will go with. So this is called segregation. Segregation is both the characters will be segregated and they are not dependent on the other factor. दोनों separate होंगे, means this one responsible for the shape of the seed and this responsible for the color. Both of them will get segregated and they will show independently. While in linkage, they do not get segregated. It means the color of the body, the yellow body and the brown body, and the, you can say, the shape of the eyes, okay, the white and yellow, that does not get segregated. Okay, so parental combinations are seen at the most, and it's very least that it shows recombination. Now what is recombination? This is linkage. Now what is recombination, children? Recombination is, okay, or let me tell you that in what conditions we see linkage. That depends upon the strength. Suppose this is the chromosome. Suppose this is the chromosome. Okay, and here these two are present over here. Suppose this is the yellow color, yellow body, and this is white-eyed. This is the, you can say, the eye color, and this is the body color. Okay, so it depends on the strength, it depends upon the distance at which they are present. The closer they will be, the least will be the chances of parental combination. And if they are further apart, suppose there is a chromosome, one is present over here, the other is present over here, yes, there are chances for it to get segregated, this will get segregated, and new parental combinations can be seen. Okay, so that can be present over there. So this is what is a recombination. So recombinations can be this way, and then when we have the linkages, linkage, when it goes together, and combination. Okay, so these are the things which happens. Okay, so hope you must have understood. I'll take the text and I'll try and make you understand. Just you can open your books and you can see. Okay, linkage and recombination. Just seeing the page 83. Okay, 83 you see there's an experiment and there it shows cross A and cross B. For example, he found that the genes white and yellow eye were very tightly linked and showed only 1.3%. So the closer it will be, like 1.3% it says, 1.3%. So the closer it will be, the less will be the recombination chances, and the further it is, the more there will be chances of getting recombination. The same is the experiment which is shown over here. Okay, the linkage experiment, you can very well understand over here. You can see the small y and small w, that characteristic. Okay, it's very close, hence recombination, the chances of recombination is very less. While the cross B you see, white miniature type and wild type, there both the genes are quite very separate. Okay, the distance between them is quite large, and hence there are more chances of recombination. Okay, so this was linkage and recombination. Hope you must have got it. Okay children, so open your textbook and go through the video. You will understand, I hope. Okay, and again, as always, you can ask me if you have anything to ask. Okay children, and do see it, share it with your friends, like it. Okay, and for the new people, subscribe.